Bismillahirrahmanirrahim. In today's PowerPoint presentation, we will be talking about the process of the gametogenesis. That is the formation of the gametes. In male, they are known as the sperms, while in female, they are known as the uh, ova, right? The formation of sperms is known as spermatogenesis, while the formation of ova or egg is known as the oogenesis. First of all, we will be discussing the process of the oogenesis. That is the process of differentiation of the oogonia into mature oocytes. You will know shortly what is meant by the term oogonia. This process basically begins before the birth of the female baby. These are the terminologies that will be bothering us in the upcoming slides, and they are basically the cells which are the first cell, which is basically a stem cell. In other words, it is known as the primordial germ cell. It will be converted into the oogonia, then into the primary oocyte, with its cover of follicular cells known as the primordial follicle, then the primary follicle, secondary follicle, graffian follicle, and finally the final product, that is the mature ova or the mature oocyte, right? So all these will be discussed step by step in the upcoming slides. Now, the morphological changes that occurs during the maturation of the gametes, first of all, the primordial germ cells, they move from the epiblast towards the yolk sac, right? Initially, when the embryo is two-layered, first layer, the above one, which is known as the epiblast, and the second layer, which is below the epiblast, is known as the hypoblast. So there are specific cells which are stem cells meant for the production of the ova. Ultimately, they reside within the epiblast layer, right? From the epiblast layer, they move towards the yolk sac, and the era is the second week of intrauterine life, right? So during the second week, the first thing which occurs is that the primordial germ cells, they move from this layer, the epiblast, towards the yolk sac. Then from yolk sac, these eggs move towards these primordial germ cells, they move towards the developing gonads, right? That area which is in future forming the gonads of this female, the future ovaries, the primordial germ cells, they move from this yolk sac towards the area where the gonads are going to be developed. And this is the time is fourth week of intrauterine life, right? So during the second week, primordial germ cells move from epiblast to the yolk sac, and then during fourth week of intrauterine life, the yolk sac, the cells from the yolk sac move towards the developing gonads. When they are arrived there at the area of future ovary, they differentiate into cells known as the oogonia, right? In all these slides, the names of the cells, they will be shown in this blue color, right? When one form of cells change into other form, you may be able to recognize, right, from this large number of front, and from this blue color, light blue color, that one form of cell, it is now converted into another form of cell, right? This is the diagrammatic illustration of what we have studied in the previous slide. This is the embryo, two-layered embryo. This is the epiblast layer, and this is the hypoblast layer. These are the primordial germ cells, and they will start from here and will eventually move towards the yolk sac in second week of intrauterine life. This is the yolk sac, right? This is developing embryo. This is advanced stage of embryo. And here you can see this is the yolk sac, and in the wall of the yolk sac, the primordial germ cells, they have been arrived here, right? Now, During fourth week of intrauterine life, from the yolk sac, these cells will move towards the area where the gonads will be developing in the future, right? These two areas, they are known as the gonadal ridge, right? And they will eventually lead to the formation of the gonads, right? So these primordial cells, they move from the yolk sac towards the area of the developing gonads. This is the fourth week of intrauterine life. And as these cells, these stem cells of the future ova, they reach here, they are given another name, right? That is the oogonia. Now, during the third month of intrauterine life, the oogonia, they undergo mitotic division. Groups of oogonia, they are surrounded by cells derived from the surface of the epithelium of ovaries, which are the follicular cells, right? Follicular cells, they are the cells which are derived from the surface epithelium of the ovaries, and they surround the groups of oogonia, right? And this is the time is the third month of intrauterine life. Then these oogonia, they may undergo mitosis to form similar cells like themselves, or they may undergo meiosis to form primary oocytes, right? Primary oocytes are next type of cells in this lineage that we will be discussing in a while. Then during the 7th month of life, all the oogonia, they have eventually converted into the primary oocytes, right? The primary oocytes, these are the cells which are resulted from the oogonia as a result of repeated mitotic cell division. I repeat, they are derived from the oogonia as a result of the mitosis, right? Mitosis. And these cells will have 46 number of chromosomes. But what happens to the primary oocyte is that they eventually they will undergo cell division, that is the meiosis. They will enter into meiosis, but in meiosis they are arrested in the prophase of meiosis 1, right? They are arrested in the prophase, which is the first phase of the meiosis 1. All the primary oocytes are individually surrounded by the follicles, right? These are the events which are happening during the 7th month of intrauterine life. The first thing which happens in this era is that all the oogonia, they are converted into primary oocytes and arrested in prophase 1. The second thing which happens during the 7th month, is that all the primary oocytes, they are not collectively, not in groups, but all of the cells, all the primary oocytes, they are individually surrounded by the follicular cells. You remember, these are the cells which were derived from the surface epithelium of the ovary, right? The primary oocytes, with the surrounding flat follicular cells, it is known as primordial follicle, right? So primordial follicle, it is nothing else but a primary oocyte surrounded with flat follicular cells, right? When the primary oocyte is surrounded by flat follicular cells, this thing is collectively known as the primordial follicle. In this diagram, you can see it is an hematoxin and eosine stand slide, in which this is basically the slide taken from the ovary, right? And in this ovary, you can see this is the oocyte, and it is surrounded by flat cells, right? These are the flat cells, the squamous epithelial cells, and this structure, this is known as the primordial follicle. Primordial follicle, and ovary is filled with thousands and lakhs of these primordial follicles, right? Eventually, this stratified epithelium, it will be converted into this simple epithelium will be converted into stratified epithelium, right? and this is then given another name that we'll be discussing in a while, right? So far, we have discussed that during the second week of intrauterine life, the primordial germ cells, they migrate from the epiblast to the yolk sac. Then during the fourth week of intrauterine life, the primordial germ cells, they migrate from the yolk sac to the developing gonadal area. Now, these cells are called oogonia, right? The first name, the primordial germ cells, then the second name, the oogonia. Then during the fifth month of intrauterine life, the number of these germ cells, they reach their maximum. That is 7 billion, 70 lakh, right? Then during the seventh month of intrauterine life, all the oogonia, they have been converted into the primary oocytes, right? Through meiosis, but they are arrested in prophase 1, right? These cells, they are arrested in the stage of meiosis 1, that is the prophase 1. Then during this 7th month of life, all the primary oocytes, they have surrounded individually by follicular cells, hence given another name, that is the primordial follicle, right? So the cells, the first cells, they are named as primordial germ cells, then the oogonia, then the primary oocytes, and then the primary oocytes, when it is surrounded by follicles, it is known as the follicle, primordial follicle, right? At birth, the primordial follicles, they are basically arrested in the diplotin stage of prophase 1 of meiosis. They remain as such until puberty, right? Until puberty, these cells are arrested in the prophase 1 of the meiosis 1. So this stage lasts from for years, right? At least 12 to 13 years, right? From menarche until the menopause. At birth, the number of these primordial follicles, they are from 6 to 8 lakh. Then at puberty, the number of these cells, they decline, right? With time, apoptosis occurs and the cell die. These cells, they decrease in number to 40,000. Among these 40,000 cells, only 500, they will be ovulated in the whole life, right? Per month, 15 to 20 cells from this pool, they are selected and they are recruited for ovulation. And eventually one of these, they will be finally ovulated, right? Now, at puberty, the maturation of the oocytes, that continues each month. What happens is that a pool of 15 to 20 primordial follicles, they are selected, or we can say recruited. All of them passes through these stages, right? At the beginning, we have primordial follicles. These primordial follicles will be converted into the first type of the second type of follicles, that are the primary follicles. From primordial follicles to the primary follicles, then from primary to secondary follicles, and then from secondary to tertiary follicles, right? Primary follicles are also known as the pre-antral follicles. Secondary follicles are also known as the antral follicles, while the tertiary follicles, they are also known as the graffian follicles, right? I will show you in a while all of these types of follicles. You will be understand what are the difference between these individual follicles. This is the cross section of the ovary. This is the fallopian tube. Blood vessels are coming and going through this tube, and in this ovary, the different types of the follicles, they are shown, right? From birth until the puberty, we have lots and lots of primordial follicles, right? In few slides back, we have already discussed that the number of the primordial follicles at puberty, it is in the range of 40,000, right? And then from this pool, 15 to 20 follicles that is selected per month during each menstrual cycle, and they will eventually pass through these stages, and finally one of that 15 to 20 follicles that will be ovulated, right? So this is the primary follicle, And so this is the primordial follicle. And how we know histologically that this is the primordial follicle, because uh, uh, a single cell is present in the center, and it is surrounded by follicular cells, which are basically simple squamous in shape, right? Then the primordial follicle change into primary follicle. And primary follicle, in histology, we recognize it as the cell. This is larger than the primordial follicle, right? The oocyte, the primary oocyte, is larger than the primordial follicle, and the surrounding cells, they are changed from simple squamous epithelium to stratified cuboidal epithelium, right? And then primary follicle will develop into antral follicle, right? Antral follicle or secondary follicle. And antral follicle is recognized by this antrum, this fluid filled space or antrum, right? Because of this fluid filled space or antrum, they are known as the antral follicles, right? Or in other words, the secondary follicles, right? This is the antral follicle. This one is the antral follicle, and this one is the mature follicle, right? Mature antral follicle, which is going to become the graffian follicle, right? And then it will eventually rupture, releasing the ova. The leftward thing, this is known as the corpus luteum, right? Corpus luteum. Now, from primordial follicle to the primary follicle, as we already have discussed, the oocyte, it grows in size, and the surrounding simple squamous epithelium of the follicular cells, it is converted into the stratified cuboidal epithelium of the granulosa cells, right? Now this is known as the primary follicle. The primary follicle is nothing else, but it is different from the primordial follicle because of the large size of the primary oocyte residing within this follicle, and because of the stratified cuboidal nature of the surrounding follicular cells. Here you can see this is the primary follicle, and this is the primordial follicle, and this is the primary follicle. This is also the primary follicle. The secondary follicle, which is also known as the antral follicle. Within the primary follicle, fluid-filled spaces, they appear and finally coalesce with one another to form the antrum, right? The antrum is that a single fluid-filled space within the primary follicle. And now this follicle is known as the secondary follicle. This is the antrum, the fluid-filled space, which eventually enlarges. And this is the antral follicle or secondary follicle. Then the tertiary follicle, also known as the graffian follicle, right? Only 1 out of 15 to 20 follicles, they will reach full maturity. When the secondary follicle is mature, luteinizing hormone surge causes the meiosis 1 to complete, right? Due to the surge of luteinizing hormone, the cell, the primary oocyte, which was arrested from the 7th month of life until now, until the release of the ova, it was arrested in a stage of prophase 1, right? Of meiosis 1. The meiosis 1, it finally completes and results in 2 daughter cell formation. The first cell, which is large in size, is known as the secondary oocyte, while the other cell, which is smaller in size with negligible amount of cytoplasm, it is known as the first polar body. The cell enters the meiosis 2, right? Now this oocyte, which was initially known as the primary oocyte, is now known as the secondary oocyte, right? But this cell, it enters the meiosis 2, but it is arrested in the stage of meiosis 2, which is the metaphase 2, right? Follicle now is known as the graffian follicle, right? Here you can see this is the secondary follicle, and this is the tertiary or the graffian follicle. And the only difference between these two is that This is slightly bigger, more mature, and the cell that is the oocyte. In secondary follicle, the cell is known as the primary oocyte, which is basically present in the prophase 1 of the meiosis, right? While in graffian follicle, the meiosis 1 is completed and meiosis 2 is started, and now this cell, it is arrested in the metaphase 2, right? Eventually, this will release, and the OVA that will be liberated, right? The OVA is finally released, and this is still arrested in the metaphase 2. And this metaphase 2, this metaphase, basically, it will end the meiosis 2. It will eventually end only and only if fertilization ensures, right? If no fertilization occurs, it's this cell, it will die, and will be shedded along the menstrual blood and the other secretory products. Now, as we have completed the oogenesis, now we proceed towards the spermatogenesis. Similar to oogenesis, which was the process of formation of the ova or the female gamete, the spermatogenesis is the process in which the spermatogonia matures into the spermatozoa, right? Spermatozoa, or the sperms, they are basically the male gametes, right? Unlike the oogenesis, which starts before birth, the process of oogenesis, which starts before birth and continues until puberty, the process of spermatogenesis, this starts basically at puberty, right? At birth, the male infant, they have sex cords in their testes. Unlike adult male, they don't have sex cords, but instead they have tubular structures, which are known as the seminiferous tubules. In place of the seminiferous tubules, the male infant, they have the sex cords in their testes, and primordial germ cells, which are the stem cells for the production of the testes, for the production of the sperms. These cells, they can be seen as large pale cells surrounded by supporting cells, right, within the sex cord of the testes. And these supporting cells, they are known with the other name of Tertiary cells. At puberty, the sex cords, they are converted into seminiferous tubules by acquiring alumen, right? And at the same time, the primordial germ cells, they are converted into spermatogonia. This is the second form of cells, which are basically the sperm stem cells, and they are the spermatogonia. From time to time, cell emerges from the spermatogonia, provides cells for spermatozoa production, right? Spermatogonia will be present all over the life, right? And they will produce the spermatozoa from time to time. Now, few mitotic division occurs to maintain the pool of the spermatogonia, right? And then if the spermatozoa is going to be formed, or spermatids are going to be formed, they undergo meiotic division, right? So mitotic division is a division that occurs within the stem cells of these germ cells, and the only function is to maintain the pool of the stem cells, right? So the spermatogonia, they are first converted into type A spermatocytes, right? They are almost similar cells, right? With some differences. Then the type A spermatocytes are converted into type B spermatocytes, and type B will eventually form through mitotic division the primary spermatocytes, right? So until now, only mitotic division has been occurred, and this only increases the number of the man cells, right? The mother cells. Then meiosis 1 occurs, right? In the primary spermatocytes, and it converts it into the secondary spermatocytes. Then meiosis 2 occur in the secondary spermatocyte, and it will lead to the formation of the spermatids, right? In this diagram, you can see this is the type S spermatocyte, which has been emerged from spermatogonia, right? This type A spermatocyte, it will lead to the formation of similar cells, which are known as the intermediate spermatogonium, right? Which are similar to type A spermatocyte, and they will lead to the formation of similar cells, type A spermatogonia, right? And they are for maintaining the pool of the stem cells, right? And then some of the intermediate spermatogonium, they will eventually lead to the formation of the type B spermatogonium, and they are dedicated for the production of spermatids eventually, right? So type B spermatogonia, that will be converted into primary spermatocytes through mitotic division, right? Until now, only mitotic division has been occurred. Then in primary spermatocytes, the meiosis 1 occurs, right? Meiosis 1 occurs in primary spermatocytes, and two cells, they lead to a formation of four cells, right? One cell lead to a formation of two cells. From one single parent cell, two cells are formed, and then from these two cells, further two cells are formed, right? So one parent cell leads to a formation of the four daughter cells, right? Type B spermatogonium leads to the formation of primary spermatocytes through mitosis. Then meiosis 1 occurs in primary spermatocytes, lead to formation of the secondary spermatocytes, and then meiosis 2 occurs in the secondary spermatocytes and forms the spermatids, right? Spermatids are cells with haploid number of chromosomes as compared to the primary spermatocytes, right? And these cells, they are connected through their cytoplasms because the cytokinesis has not been completed yet. Then from this step onward, a process known as the spermiogenesis occurs. And this will lead to the formation of the spermatozoa, which is the final product. The spermatids are surrounded by the supporting cells known as the Sertoli cells and the leading cells. Pituitary hormones, the LH and the FSH, they act on these cells and are necessary for the testosterone production and the spermatogenesis. The spermiogenesis, as already discussed, this is the series of changes that results in the transformation of the spermatids into the spermatozoa. It is known as the spermiogenesis. It includes acrosome formation, right? Acrosome is the basically it is the lysosome-like thing present at the tip of the sperm, right? And we will discuss in fertilization topic that it is needed for acrosomal reaction, right? Which is one of the important reaction for the fertilization. Then the second process that occurs after the acrosome formation is that the nuclear material that is condensed, right? Then the tail, nick piece, and mid piece, they are formed, and the excess of the cytoplasm that is shedded, right? Here you can see this is the spermatid, and this is a cap of acrosome that is forming. So the acrosome, you can see this is forming gradually, and eventually it will cover the whole of the head of the sperm like the helmet, right? So this is the acrosome formation, the first thing that occurs during the process of spermiogenesis. The second thing is that the nuclear material, it condenses, right? Here you can see the nucleus is big in it is larger in amount, it is bigger, and eventually the size of the nucleus, it is decreasing, and the nuclear material within the nucleus, it is condensing. Then the tail, it is eventually forming, right? Similarly is the neck and the mid piece. And the final process is the shedding of the excess cytoplasm, right? You can see this is the cytoplasm. This is the cytoplasm, which will eventually decrease in size and then shed it off, right? And the final product, which is a sperm with a proper head, neck, mid piece, and a flagella-like tail. This is known as the final product, the sperm. This is the end of the slideshow. If you have any sort of query, you can ask me in the comment section below. Thank you very much.